Welcome to For Whom the Cell Tolls. I'm your host, Keenan, joined again by Scout Pomeranian and Ranger GSD, both little adoption dogs. You can kind of follow their story if you follow our stuff, if you're new here, that is. But anyway, been a little bit, I understand. Teaching's hard work, so I end up sort of ignoring the fun science side of things and the podcast sometimes. But, we're back, and with some renewed energy. We're in the middle of the semester, so it's fun. Okay. Big deal, too, because the last episode I left on was talking about the ASH meeting and some new stuff that's happening. But today I want to address some controversy in the field. This is going to be a fun one, I think. Here's the deal. One of the cool things that we have in the lymphoma field is, like we've shown in other episodes, Chimeric Antigen Receptor T-cells, where we take a patient's T-cell out, we genetically modify it to hunt the tumor, we send it back in, and it chews them up. And to provide some context again here, if you cross and you fail the initial chemo and immunotherapy of R-CHOP in the lymphoma we studied, diffuse large B-cell lymphoma, if you fail that and you don't be one of the 50% of people or so that has a long-term remission or cure, is what we can call it, you faced, back in 2017, something like a 15 to 20% chance of making it 1.5 to 2 years or so. CAR-Ts come along. That number, that 2-year and 1-year number, is now closer to 50 and 60%. And it's so good. These CAR-Ts are so good. We have plenty of physicians and scientists that are essentially promoting, what if these were first-line treatments? Now, that doesn't get into the cost and what we call financial toxicity and some of that stuff. Part of that's why this episode's happening as well. But key thing, and I'll just tell you this as a PhD person, a scientist, these things are a paradigm shift. They are, I don't want to use the word miracle lightly, but it's a big deal. Nobody sees shifts like that without making a big difference. Given that, there is a lot of chatter, and you can go into the darker corners of the internet, and you don't even have to go too dark. You can go to some pretty popular podcasts on this platform that have started to have a bit of a misguided interpretation of what CAR-Ts are. Right now, instead of treating them as this cancer therapy that we can control, and we do, there are plenty of voices and microphones out there that have gotten a little riled up that you won't put that cell in me, that cell's going to kill me. How dare you? Nobody's going to put any cells in me, that they're trying to kill us, they're trying to control us. I do think that since COVID and, mRNA vaccines, which deserve their own episode for their role now in cancer vaccines, I think people are a little more hungry for, everybody's out to get you type of stuff, I'd say. So you don't have to go too far on the internet to find people that are really upset that CAR-Ts have become mainstream. If you've been listening to this podcast a long time, like I'd say the 37,000 of you that do, you know that this has been a long road. It's been a very cool road too. And like I said in the beginning of the episode, you either do the CAR-T or you face the 10%, 20% chance, right? Now, given all this, it is totally okay to be afraid of something that you haven't encountered or don't understand before, okay? Hey, I didn't invest in stocks or anything. I was terrified of, economics because I'm kind of an idiot like that. That's okay. But it doesn't mean I should say that it's all a fraud, right? Now, given that, let's go through something that did and has been showing up. Since CAR-Ts have becoming mainstream, there's been a few cases of secondary cancers. And this is definitely troubling people. What a secondary cancer is something that comes back, but it's a different cancer than the original one. This is surprisingly actually a pretty big consequence of just regular old chemo and radiation, right? So if you go in with a tumor, and maybe it's a tumor type that responds well to chemo radiation, and, plenty of patients get cured with that. But then later, maybe 5-6 years later, a new type of tumor pops up, and we have done studies enough before that it's like, oh, dang it. The chemo ripped apart the DNA during the cell's S phase so bad that a few cells actually survived in a different tissue and became cancerous because of those mutations. Dang it. And that's always been one of the stranger, double-edged sides of chemo and radiation and anything really that hurts DNA, right? Or twists DNA or makes chromosomes different, anything, right? So secondary cancers have always been around. But where this comes up with the CAR Ts is that you put a new cell in to somebody. It's their T cell, but it's supercharged. It's very new. A few cases were reported that after treating B-cell lymphoma cancers, a few T-cell cancers showed up. Something around the edge of like 23 or so. And it was mainly reported, and like at least it was more officially reported last year, December 2023, by Verdun and Marks from the FDA. About 23 cases of post-CAR T T-cell lymphomas. Let's just say that this did cause a bit of a stir at the ASH meeting. A lot of presentations began with, we know there's a couple cases out there with this. We're looking into that, but statistically, it's tiny. They're correct on that. Think of the thousands of people that have been treating with this. And I mean, like maybe even hundreds of thousands. Maybe my numbers are too small. But to report 23 is a tiny fraction of that number. Here's the other thing later, because we're going to talk about this episode. That FDA paper that I'm talking about, it gets pulled later. And now if you search it on the internet or when it's cited by the two papers that we're going to talk about today, or the one paper, you can't find it anymore. Because essentially what these papers we're going to talk about today, or the main one, the Ash-Alizeta one, they sort of showcase, okay, this is not where they're coming from. But like I said, this was going to get a little hairier, right? I told you that would happen. But like I also promised, you wouldn't, if you were a patient faced with this and CAR Ts were on the table as a treatment option and the other options were more of a 10 to 20% option, and this is a 50 to 60% option, I got to tell you, like, if I'm trying to live longer, I'm probably putting the CAR T, putting them in me, basically. That's all I can say. So a few of these cases pop up, and remember, social media takes off kind of like a virus, and you've got plenty of people being like, this is causing the cancer. They're killing us, right? And immediately that's where this goes. So people get their stuff together. People at some of these bigger hospitals have been treating these patients a lot longer, et cetera, et cetera. They go back in time and say, okay, how many of these patients or how many of these cases have this secondary tumor, right? Well, that is essentially what Mark P. Hamilton, first author, followed by senior authors Ash-Alizeta and David Miklos from Stanford and one of the major hospitals in the Netherlands, basically set to find out with a large Stanford cohort. So, among 724 patients that received CAR infusions, 96% were CAR T-cell infusions. There are other CAR-type cells you can do, but still. Of those 723, 14 did develop secondary hematologic tumors. 11 others developed solid tumors. The 11 solid tumors are a pretty good case for like, okay, you treated things pretty hard. You probably made some DNA decisions in there. They might have triggered something. Here's the other thing you got to remember about the statistical population you're working with. Most lymphoma patients are pretty old. And if they got one cancer, they could totally probably get another, right? So you always got to take that into account. Next, let's take a look at these 14 blood cancers that did pop up. Of the 14, 13 of them were myeloid cancers, macrophage type things. Totally off in a different evolutionary spot, but they will sort of play into the major hypothesis that the paper builds on. One of these 14, so one of the 724 that were treated, did develop a T-cell cancer. So what does that mean? Immediately the thought would go, those are the CAR T-cells becoming a cancer. And I'll tell you a little bit why people maybe have thought about that. So I want to bounce to a secondary story to build on the Alizeta and Hamilton and Miklos paper to another one. Julia Garcia, the first author from UCSF in 2024, documents a new CAR T making protocol. Here we go. So normally what you do with a CAR T is you send in a viral vector that will integrate DNA. That DNA has the genes necessary for the CAR T to find and kill its target. Usually that's going to be an oncogene like CD19, CD20, or an oncomarker that is going to allow the CAR T to hunt and kill that tumor. Amazing. What we've been doing in the last few years is adding power to these CAR T cells. We add cytokine receptors, we add co-stimulatory receptors like CD28, all this big stuff, right? The Garcia paper went a step further. They wanted to make a super mutant CAR T cell. They went ahead and added a gene that causes the CAR Ts to grow, proliferate, and survive. Those are 3 hallmarks of cancer right there, right? But if you're making a predator, you want it to be strong, right? This is the natural direction, but it's a strange reversal, right? The gene that they added in, I think they did another one, but this is the main one, a gene called CARD11. It is strictly an oncogene in DLBCL tumors. They added it in there to these T cells because it forms this massive CBM signalosome, this big internal signaling mothership gene, and it causes these cells, whoever has it, to not die, to proliferate, and to survive. Perfect, right? And you want that for your CAR Ts. You don't want them to get exhausted. You want more of them, right? So the crazy thing is that we've come so full circle, is that we are adding oncogenes to the tumor hunting genes to make them stronger. I used the oncogene to kill the oncogene, right? Little Thanos reference there, even though it makes me sound old at this point because that was 2018. But the good news is, like, even though the Garcia paper is super cool and a little bit scary, they have a whole section talking about, hey, we did add in a kill switch for these cells. We did add in some escape mechanisms for these things in case they get out of hand. They have a specific, I can't remember what they said. It was like a specific DNA chop mechanism that is gonna like be activated maybe by something like tamoxifen, similar to how like Cree works. But it was still full circle to see the thing basically use an oncogene and make these CAR Ts more powerful. Papers like that are where people are gonna say, what in the world are you doing? How dare you? But in our world, like, you got to make them last, right? Okay, transitioning back to paper number 1, the Ash-Alizada, Hamilton, and Miklos paper. Let's go back to our single little T-cell tumor that they found among the 724. How can we determine, is this a tumor of CAR T-cells? So, in the T-cell tissue, some of the DLBCL, the original disease, a tiny fraction of that remained. It had a TET2 mutation and a DMMT3A mutation, suggesting something called clonal derivation. Highly clonal T cells, stem-like T cells. Because the neat thing is that B and T cells share a common ancestor. Before a B and T cell ever exist, there is just one cell in the hematopoietic stem cells in the bone marrow that can become any of them. So the cool thing is that technically what they end up finding is that this T cell tumor and a lot of the ones that had the myeloid issue, they were actually there with these same clonal mutations that eventually would trigger the DLBCL. And the DLBCL just showed up more. Typically, B cells undergo a bit more of an aggressive, somatic hypermutation, all that crazy stuff. So they are the ones that take those initial mutations and run with them and become a big tumor, an anger, basically, right? So when that B cell tumor shows up with those mutations, we send in chemo, it doesn't work. We send in the CAR T, it works. It eliminates them, right? What that did is that there was a stem cell that had the TET2 and DNMT3A mutations all the way back in time that didn't die from the CAR T cells. That would be allowed to become an immature T cell that would eventually become a pre-malignant clone near the thymus when it would basically try to make a CD4 to CD8 commitment. With the B cell clones and tumors completely deactivated. This patient also had an EBV infection, Epstein-Barr virus. That was enough with the CAR Ts to basically cause this T-cell lymphoma to pop up. So, from this point, We also have to prove that this T-cell lymphoma is not from the CAR Ts that you infused, that it is actually an ancient stem cell that has found its way through selection. So the next thing that they do, okay, we're going to measure for any transgene RNA from the vector from this T-cell lymphoma. They get in that T-cell lymphoma, they check it out. There is no CAR RNA, which means there's no gene expressing it. They go a step further. They sequence these for all their DNA and say, is there any DNA integration from the original viral vector plasmid that we gave the CAR Ts? If there was, it would indicate that this tumor was from the CAR T. When they sequence it, nothing. Clean. It's just a T-cell tumor. Now, the other neat thing is that technically this patient still had CAR T-cells at a very low level running around, sort of patrolling. You want that from the product. You want them to keep patrolling for a while. But those CAR Ts were genetically distinct from the T-cell clone that became the lymphoma. Problem solved. Excellent. So the key finding, and I'll try and link both these papers, key finding essentially is that the original cause was this ancient dual possibility T and B cell progenitor. The CAR Ts killed the B cells. The TCL lymphoma rose out of that inflammation caused by the CAR Ts killing the lymphomas, right? And it didn't help. And it didn't help, basically, that the EBV virus was there as well. But hopefully this is a good little discussion into here's what it takes to, basically when you're in a new sort of therapy paradigm shifting type of thing here, we're in a whole new world of what's possible with this. And that's why it can get a little different. And at the same time, it was a very good detailed look into things. Equally, it really gave us some good biology on where some of these secondary cancers come from and how early some of these B-cell lymphomas begin. Because this kind of showed hey, some of these mutations have been hiding since before this thing was a B-cell. This gives us another good option of saying, if we can start sequencing for these founder events, or at least screening for them somehow, we could tell people that have in their bone marrow, and again, doing a bone marrow sequencing and biopsy, it's not it's off the street, easy. And maybe there's going to be a better way circulating DNA or something. But if these mutations, these ancient original mutations are happening in these progenitor stem cells, if there's a way to warn patients and warn clinicians that this is coming, we could do better screenings, we could do all kinds of great stuff. It's just really hard to find these little guys because they're so close to what the healthy cell looks like. But then again, when you have these clonal mutations, it means that in a lot of cases, TET2 deletion, DNMTA3 mutations, things like that, if we can screen for these specific things that we are starting to zero in on as to who's a bad guy, who's a bad mutation, this and that, we could possibly start sort of looking for signs that are associated with these. Perhaps there's some sort of clinical factor that changes in that case. I don't know, we'll have to see. But to kind of finish things out, the Garcia paper is probably only the beginning of adding power to these CAR T cells. And that power is going to be extremely high, to the point that essentially we're adding, like I said, we're essentially adding oncogenes to these things to make them survive and proliferate. It will not be the first time that controversy will show up with CAR T. And I do sort of feel the rumblings beginning the same way towards what we will eventually be trying in the very near future, which is big cancer vaccines loaded with 30 to 40 mRNAs that are cancer alteration targets. Pancreatic cancer, for example, is having one of those tested now, I believe, because remember, pancreatic cancer is very hard to detect kind of physically until it's already spreading everywhere and the patient dies. So if you could make an immune system response to those initial genes that we know create the pancreatic tumor, it would make sense if you're high risk for pancreatic cancer to perhaps consider a vaccine like that. Now, this is not a vaccine podcast, it's a cancer podcast, but I do believe we'll eventually try and confront the mRNA vaccine future, I'd say. But for now, the CAR Ts. Here's what I'd tell you as a PhD in lymphoma. If you've got a secondary B-cell cancer and it's already overridden your original regimens, I'd go ahead and elect for the CAR T if you're eligible. Yeah, I feel like that's only statistically going to help at this point, especially since, remember, most DLBCL patients are over 55 at least. So you want to maximize your chances on these. And, the cool thing is, even though we're talking about something that's very different in terms of cancer treatment, we're getting towards the 5-year, 6-year mark from the initial populations that we started to do CAR Ts with in large number. 5 to 6 years is getting towards that C word, that cure word, okay? We're holding out hope with that. It's going to be awesome, but we'll see. And I think to end, I don't know if I've told this story on the podcast, but I read Siddhartha Mukherjee's Song of the Cell, and he talked about the first CAR T patient, Carl June treated at Penn, I believe, and how it was a girl with leukemia. She was dying. Nothing was working. He used the CAR T. She went into that nasty CRS where the cytokines fry the brain a little bit. He penned his resignation because he thought he had just killed a child. But she came out of it. And the CAR Ts cured her leukemia, and she's still alive today. So the hope is, even though there is going to be a lot of details to work through on these, I would hold out. I am a big hopeful person on this. Totally different episode if we want to talk about cost. That's not really my department as much. I can just be happy about things for now. Anyways, I'm hoping to do a few more episodes. Like I said, teaching takes up some time, but if you have friends that enjoy this or enjoy science or cancer type intrigue, share it. It's always a good time. Thanks so much for listening, and until next time.